All right, we're gonna go ahead and get started just to make sure we have enough time to get through the lecture and have enough time for journal club after. So I think the plan for today is we're gonna talk about the cytoskeleton and go up until 2:45. So hopefully that's enough time for the journal club. So if I'm rambling on and it's 2:45, feel free to just raise your hand and tell me. If there's any lecture material that we haven't covered, I'll just record that after and post it for a video to watch. But hopefully it shouldn't be too much. And if there's any questions about anything and that we can't get to, feel free to email or post on Piazza or anything like that. So I hope everybody got excited about the project, submitting their ideas for their disease, and is excited about the journal club today. So we'll get going on the cytoskeleton. So building upon some of the things we were talking about with the extracellular matrix, as well as enzymes. The learning objectives today is diving a little bit more into some of the different cytoskeletal filaments that you learned about in the pre-lecture, specifically about their polarity and the aspects that regulate their polymerization. And the disease application for today is Listeria, which is a type of food poisoning. So getting into the food poisoning caused by Listeria, it's known as listeriosis. And this is generally an infection of the GI system or the gastrointestinal system. It can influence your nervous system as well in really severe cases, but is generally thought of as causing food poisoning. And this is caused by this bacterium, Listeria monocytogenes. So this is a bacteria that generally lives in the soil. So if you play around in the dirt too much, maybe you can become exposed to this, but it becomes really dangerous if it is ingested. So a lot of times this is found in different animal products, different deli meats or soft cheeses, and becomes invasive when it infests a mammalian host. So this is a pseudocolored electron micrograph. These little things in green are what Listeria looks like. So like I said, this is generally contracted by consuming raw meat or unwashed vegetables. And this CDC link down here has information about recent foods that have caused these outbreaks in the last 10 years or so. So the incidence and prevalence of Listeria food poisoning, about 1,600 infections and 260 deaths annually are generally happening in the United States, with an incidence of about 0.3 infections per 100,000 people. So it's not crazy common, but it can be really dangerous if you get it, specifically for people with increased risk factors. So these are often people with compromised immune systems, specifically pregnant women. Is very much advised against any threat towards Listeria food poisoning, newborns that are still having a developing immune system, and other people with immunocompromised states. So for healthy people, the symptoms are generally transient. This bacteria is able to be fought off by your immune system, but just have some GI problems like diarrhea and fever, maybe lasting a couple days. For pregnant women, this can escalate to muscle aches or problems with the fetus, and other types of high-risk people. This can lead to that infection of the central nervous system, like I said, causing loss of balance or convulsions, even meningitis, which is just inflammation of the brain or spinal cord. All right, so what this actually looks like, so this is a little video of Listeria in a cell. So it kind of shoots around inside the cell and can even shoot out of the cell and then back in. So if you look like right around here, you can see some of these bacteria kind of shoot out and come back in the cell. And this is really good for this bacteria because as long as it's staying inside the cell, it can generally evade the immune surveillance. And so this will become more clear when we talk more about the immune system. But the immune system is generally existing outside of cells and responding to things outside the cells. So as long as this bacteria can just shoot from one cell to the inside of another cell, then it can go unnoticed. And so this is transmitting from cell to cell where it can divide and replicate within those cells and grow into an infection. So we'll talk a little bit more about how this does this at the end of the lecture, but as a sneak peek, it leverages the cytoskeleton to do this. So the cytoskeleton is this network of different filamentous proteins. And so this is like the structure or the scaffold within the cell that does many different types of functions. Transport, it allows structure and enables cell motility. And there's different kinds of protein binding. It allows that transmission of signals from the extracellular matrix inside to cause changes in gene regulation at the nuclear level. And this is a very complex network, similar to the ECM. Like you learned about in the pre-lecture, there's lots of different types of proteins that are interwoven, that are assembling and disassembling as they're needed to cause their certain functions. So we're going to specifically be talking a lot today about this assembly-disassembly process of these cytoskeleton proteins and how they're regulated, since this is what is leading to their function in a lot of cases. So we're going to talk about this generally. So all of these filaments are growing or shrinking by an addition or loss of monomers. So these are polymer chains, which are just long chemical molecules made up of repeating units. So these monomer units are going to be added or subtracted from these polymer chains these are protein polymers inside of the cell that are going to lead to their growth or shrinkage. So the growth is therefore, it's referred to as polymerization because it is the forming of a polymer. So as you're adding more monomers, you're increasing polymerization. And a couple broad categories that are going to be regulating this extent of polymerization. Predominantly, the concentration of monomers is a big one here. So this is similar to when we talked about enzymes, we assume that there is always a lot of substrate around that can bind the enzyme and react. And inside of the cell, this is very much not the case with these monomers. There's a limited amount of them. And so the availability or lack of availability of the monomers to bind to these growing cytoskeleton chains is going to be regulating whether they grow or not. But there's also different accessory proteins that are acting both on microtubules as well as microfilaments that are going to be controlling this process and regulating the availability of these monomers. And phosphorylation is another key aspect that is controlling the growth or shrinkage predominantly of myosin filaments and intermediate filaments. So we'll talk more about these in the later slides. So we can think about this process of polymerization as occurring in multiple phases. So if you have a cell, say maybe there's no cytoskeleton, there's just a bunch of these monomers available, they'll start by just bouncing around off of each other and eventually forming these dimers or trimers. And this start of the polymerization chain is known as nucleation. So nucleation doesn't have to refer to a polymer chain or a cytoskeleton filament starting from nothing. It can be any kind of new growth of a polymer chain. But this initiation point is always called nucleation, and this is the beginning of the growth phase of this polymer. And then all these other free monomers that are in the vicinity will continue to bind and grow this chain, increasing polymerization. And this generally happens fairly rapidly. And this phase is known as elongation. So as you can see, there's a little bit of lag before this nucleation process occurs. But once that occurs, elongation occurs very rapidly. And then this monomer pool is fairly depleted, and so these polymer chains reach a steady state where there's some monomers being added, some being subtracted, and that leads to kind of the same concentration of monomers in the surrounding, as well as a general stable length of the polymer chain. All right, any questions so far about this? Okay, so getting a little bit more specific, we can define what this steady state length is by defining something called the critical concentration. So looking at this in a similar way as we did the enzyme kinetics, we can write this as a rate equation where L is the length of our polymer chain, and the change in the length over time will be a function of how many monomers are added and how many are subtracted. So this rate of addition of monomers is a function of the concentration, and the rate of subtraction is not. It's more about the stability of the polymer chain. But if we assume this to be at steady state, so we're looking for what is the steady state concentration. And so we can set this rate equation equal to 0, do some algebraic manipulation, and we can define this concentration of monomers at steady state to be the ratio between the on rate of monomer addition, or the off rate of monomer subtraction, to the on rate of monomer addition. So that's K off over K on. And so similar to the Michaelis-Menten constant, we can define this as Kc and refer to this as the critical concentration. So this refers to the concentration of monomers at which this polymer chain is at steady state. Okay, and a reason why we would want to do this is because it becomes a really useful tool functionally. So if you think about the concentration where this polymer chain is at steady state, if you have a higher concentration of monomers than that, then your filament will grow, or you can increase polymerization. There's more availability of monomers around to add to this growing filament. But if your monomer concentration is below Kc, or this steady state monomer concentration, then it'll lose monomers and it'll shrink as a result. So this Kc is really important to define because this, similar to enzymes, is an inherent quality of that monomer chain. And there's different things that can happen to filaments that will change the Kc value. So it'll change whether or not it would be favorable at a given concentration for monomers to be added or subtract. And this is part of the way that these filament growth and shrinkages are regulated. Yes. Exactly, yeah. Steady state monomers are still adding and coming off, but it's just at the same rate. Exactly. Yeah. Any other questions about this? Yes. Yeah, so the K on is the rate at which the monomer is being added. So this is a rate constant, and the K off is the rate constant that determines the monomers coming off the filament chain. Yes. So are you referring to the M? Yeah. Yeah. Yeah, good question. So this first equation right here is just the rate equation that we would write to define how this length is changing. So this is going to be a function of how they're added and how they're removed. But moving to the second equation, we're going to assume that this is at steady state. So when it's at steady state, this length is not changing as a function of time, And so we can set this to equal 0. And so that's where this SS subscript comes in. Thank you for pointing that out. I should have explained this. This SS just is referring to the monomer concentration at steady state. Yes. Yeah. So eventually these polymers are always going to reach steady state as long as this Kc is not changing. So this is generally how these filament growth and shrinkages will be regulated. And we'll talk about that more specifically, what that looks like with different types of filaments. All right, thanks for the questions, everyone. In terms of microtubules, so this slide might look familiar from the pre-lecture. So these are protofilaments that are rolled up into groups of 13. So around this circumference here, there should be 13 of these, and these, the tube-like structure or the protofilament rolling onto itself like this creates a really rigid structure. So this these microtubule filaments have a lot of strength to them, which is important for the function that they play in the cell. And similar as what was mentioned in the pre-lecture, these are really important because they have some polarity to them. And so this is going to introduce some directionality to these filaments that allow the transport of things towards one end or another. So if you can imagine, you have some protein that you want to get out of the cell, you need to be able to know what direction to travel in on this cytoskeleton network to get you closer to the outside of the cell. So this directionality is important for transport within the cell. And we can think of these as the plus end and the minus end. This nomenclature is fairly arbitrary, but we can remember the plus end because this is also generally the end that the microtubules are growing at. So these microtubules can grow at either end, but in cells, this is preferentially occurring at this plus end, which refers to where the beta portion of this tubulin dimer is exposed. Yes. So we'll talk about this more when we get into molecular motors, but there's certain types of molecular motors that are proteins that carry cargo and they walk along these microtubules. And so they respond to the different monomers in this tubule chain by going in one direction or another. So there's different types of molecular motors that will go either towards the plus end or towards the minus end. And these will play specialized functions in terms of where things need to go in the cell. Good question. Okay, so in terms of the microtubule growth, like I said, this is happening preferentially at the plus end. And something that can be confusing that I just want to clarify here, they're made up of these alpha beta dimers, but these alpha beta dimers are going to associate with each other before being added onto this growing filament. So when we talk about the growth of these filaments, we'll refer to this alpha beta dimer as a monomer. And this is just because they're being added as one unit onto this growing chain. Okay, so this is where that becomes important because you can see in this schematic, now the coloring is a little bit different and we'll talk about this, but these groups of two are being added together. And so this is how we can refer to the growing of these microtubules is the addition of these alpha beta monomer units in light of the whole polymer. But something that is really cool about microtubules is that they're also enzymes, and specifically they're GTPases. So that means that they cleave GTP. And when they cleave GTP, it becomes GDP. So what's happening in the growth of microtubules is you have these tubulin alpha beta monomer units existing in the cell, and then they're going to bind to GTP. And when they're bound to GTP, then they will come and preferentially bind to the plus end of these tubulin chains. And so this GTP allows the conformation to fit in well to the addition of this chain. But because this is an enzyme, eventually that GTP will hydrolyze to GDP. So once the alpha beta unit is bound, then the activity of this GTPase enzyme is increased, and this GTP is hydrolyzed, meaning the one phosphate group is cleaved and it becomes GDP. So this is guanosine triphosphate and guanosine diphosphate. So what this leads to is this, what's known as this GTP cap. So because all of these GTP bound monomers of tubulin are binding to one end, it creates this section of the tubulin, microtubule that has all of this GTP bound. And this is preferential for more GTP binding. And this is because it lowers the Kc, or the critical concentration that we just talked about. So this is one way that it's regulating growth preferentially at this plus end, is that as long as there is these tubulin monomers with GTP bound, or this GTP cap specifically, it is preferentially going to bind more. So that means that there needs to be a lower concentration of these monomers present to continue to grow this polymer. Does that make sense? So let me just explain it one more time, because this can be confusing. So if there was no GTP bound, there's one set critical concentration, or Kc. But before the GTP is hydrolyzed, when it's still a GTP, or this GTP cap shown in green, the Kc is lowered. And so at a lower Kc, this means that there is a lower concentration of monomers required to maintain steady state. So if there's still the same concentration of monomers in the surrounding area and the Kc is lowered, that means there's a longer amount of time that these monomers can be added where the chain can still grow. Okay, so this GTP bound cap is going to be present as long as this enzyme activity isn't progressing. So the longer these tubulin monomers are bound, the more likely it is that they act enzymatically on their GTP, cleave the phosphate group, become GDP, and then the Kc is going to go back up. And so the Kc going back up means that there needs to be a higher concentration of monomers to maintain steady state. So say we're at a concentration that is below that Kc, what that means is there's going to be a dissociation of monomers. So if there ever comes a time when there's not this GTP cap, there's not a constant addition of monomers, then this GTP cap goes away because of the hydrolysis that's happening through this GTPase activity. And then these monomers will rapidly dissociate. And so this is known as catastrophe. And so this is a rapid shrinkage. And what this leads to is something known as dynamic instability. So this leads to basically, if we have one monomer chain here and this green is our GTP cap, it keeps growing and keeps growing, and then eventually there's not enough monomers to keep growing it. It reaches its Kc, then that hydrolysis occurs, it loses its GTP cap, becomes unfavorable, and then it rapidly dissociates. And so that GTP cap is gone and it goes away. Oh, I think I went too fast. That GTP cap hydrolyzes, it goes away, and then all those monomers dissociate, and then they reassemble into other tubulin monomers that are going to add to different chains of microtubules. And so this leads to kind of a constant growth and disassociation of these tubulin polymers or these microtubules in the cell. And we can visualize this using fluorescent dyes of this. So what you can see is these chains kind of growing and shrinking at all different points in the cell. Yes. This one? Yes. Yeah, so this is, say, one microtubule chain, and this has grown and grown, and then there's not a lot of monomers left in the surrounding area to continue to grow it. And so that means it'll lose its GTP cap because the enzymatic activity there cleaves it. And so when that happens, then you get catastrophe. All of those monomers disassemble, and then that increases the monomer pool, and then new microtubule chains can grow from that. Does that make sense? Okay. Yeah, so this dynamic instability or rapid growth and disassembly of microtubules in the cell is really important, kind of an elegant way to reach all the different areas of the cell. So if you just had one static microtubule filament, that would only reach to one point in the cell, but because it's growing and expanding, it can cover kind of a wider area, and every part of the cell can be reached that way. Yeah, so in terms of cargo transport, this can be really important because these are often serving as these train tracks for moving proteins and other components of the cell around, like I mentioned before. So another way that these are structured, these are kind of all coming together at one area and expanding out to cover the cell. And so these middle parts of where the microtubules are associated is known as the microtubule organizing center. And a centrosome is one example of this that's been really well studied. We'll talk about that more in the next coming slides, but this is another reason why this growth is preferentially occurring at the plus end, because the minus ends are all kind of stabilized around this microtubule organizing center, or MTOC. And there's a lot of different types of these in the cell, as was mentioned briefly in the pre-lecture. But something important that's happening at these MTOCs is they will nucleate growth, and they're stabilizing that minus end. So that allows the plus ends to experience this dynamic instability. Okay, so another kind of movie that demonstrates this is relevant for mitosis and chromosome segregation. Like I mentioned, the centrosome is a really well-studied example of the MTOC, and you can see these microtubules kind of pushing around all over the cell, searching for the chromosomes, finding them, and then pulling them as the cell divides. And so this is a major function of microtubules in the cell and becomes really important for all types of cell growth and division. So this process of chromosome segregation is greatly facilitated by these centrosomes that are a type of microtubule organizing centers, or MTOCs, and they allow the microtubules to connect to, disassemble, and divide these sister chromatids during cell division. And this is part of mitosis. So the microtubules are a component then of the mitotic spindle. And these microtubules in the spindle are linked to the centromere. So the centromere, as you might remember from biology, is this central part that links these two sister chromatids together. And these have specific binding sites for these microtubules. And this specific binding site that you'll need to know about for this class, as you can see, it's really complicated. There's a lot going on, but the kinetochore is the main component of the centromere that is binding these microtubules and enabling them to connect to and segregate them. All right, so in terms of cancer, the main treatment of cancer are these chemotherapeutics that are anti-mitotic drugs. And this is interesting because many of them are actually targeting microtubules because of their important role in mitosis and chromosome segregation. So without the proper function of microtubules, you can't have accurate cell division, and this would preferentially hinder cancer cell growth because they're undergoing mitosis at a much faster rate than any other type of cell in your body. So these are just a couple examples of different chemotherapeutic drugs. You don't have to know the name or what they target specifically, but vinblastine causes tubulin aggregation. So this just clumps up the microtubules so they can no longer polymerize. Some promote depolymerization, so that would be lowering the Kc through different means or stabilizing microtubules that blocks this dynamic instability that allows them to search for chromosomes in the process of mitosis. Interestingly, some of these were identified originally in plants. So periwinkle, for example, is a plant that would be very toxic if you ate it, as well as any of the needles from yew shrub trees or other things. And so plants can use these to block different types of growth that might be invasive, such as with fungal species or other things. Okay, switching gears, try to speed up a little bit. This is kind of a similar story, but now moving from microtubules to actin filaments. We have the same type of idea. There's still polarity in the cytoskeleton filaments, which means they'll grow preferentially at one end. And there's monomers that are making up the growth of these larger microfilaments. So in the case of actin, the monomers are known as G-actin. This is, you can think of globular actin, where the polymers are known as F-actin or filamental actin. And this is much smaller than the microtubules, much less rigid, and they're also acting as enzymes. But where microtubules were GTPases, actin are ATPases. So in both of these cases, it's interesting, they're utilizing something that is a high energy molecule to facilitate the growth of these polymer chains. So similar to in microtubules, we have a plus and the minus end of the actin filament, and the plus end is growing faster. And this is again because of the difference in Kc. And this is a function less of the enzyme activity of these monomers and more of just the way that they are oriented, because their polarity, the way the monomers assemble together, it's much more energetically favorable for monomers to be added at this plus end. And the ATPase activity works in a similar way. So in terms of, sorry, I'll just skip to these animations. So in terms of the addition of these monomers at the plus end, you have all of these ATP-bound monomers at the plus end that will eventually hydrolyze. And then if you look at the minus end, you have these ADP-bound monomers. So along with the inherent lower Kc at the plus end, the hydrolysis of ADP to ATP also further accentuates this difference in Kc. So this Kc at the plus end is in this growing chain generally much lower than at the minus end. Yes. So the monomers are binding with the ATP already attached. And when the monomers bind in the chain, that changes the protein conformation to make the enzyme activity slightly more favorable. So it's not like trapping the ATP. ATP is very small compared to the whole G-actin monomer, and definitely compared to the whole filamental actin. So it just occurs within the chain. Does that help? Thanks for the question. Okay, I think we might make time. So along with the difference in KC, there's a couple of proteins, accessory proteins, that are also regulating actin growth. And this is true also of microtubules, but these proteins are specifically thought of in the context of actin. So if we look here, these can be considered ADP-bound monomers of actin, and profilin is a protein that binds these ADP-bound monomers and facilitates this nucleotide exchange. So it removes that ADP and enables the binding of a new ATP molecule to these actin monomers that allows them to add to the growing chain. So this enhances the growth of these actin chains. Thymosin is another accessory protein, and this binds the monomers. So specifically, it binds the ATP-bound monomers. And this can be important in terms of times of rapid growth or change in the actin filaments. So you might think this restricts the growth, but what it does functionally is it creates this pool of available actin monomers that can be released at any time. So if you have thymosin bound to a lot of these ATP bound monomers, and then there's some trigger that the cell needs to rapidly polymerize actin, they can release these, greatly raise the concentration of available monomers, and that will speed up this polymerization process. So profilin also acts as a shuttle for these ATP-bound monomers and just delivers it to these growing chains. And so all of these together can work to grow the monomer or the polymer chains for the polymerization of F-actin. And these are definitely not the only accessory proteins. Like I said, there's others that are acting specifically on microtubules. But cofilin is another protein kind of like thymosin, but this binds the ADP monomer. There's galsolin, which is an actin severing protein. So again, this might think that you're depolymerizing, but when you sever an actin chain in the middle, what it does is it creates new sites for growth. So new points for nucleation to occur at different points of this growing chain. So this can actually help increase polymerization as well. And alpha-actinin and filamin are cross-linking proteins. So if you remember from the pre-lecture, actin generally exists kind of around the inside of the plasma membrane. And these types of proteins can be important for stabilizing and adding strength to the plasma membrane for these actin networks that are there. All right, so this cross-linking is specifically important in terms of cell motility. So this is just a fun little video of a neutrophil chasing a little bacteria around. And something that you can appreciate from this is the force that the cell is able to exert as it's moving in this appropriate direction. So you can see it's moving these little beads out of the way in order to make it to the bacteria. And this is partly due to this polymerization of actin occurring at this leading edge or the front portion of the cell movement and the different cross-linking that's occurring there. It's always fun to watch it get the bacteria at the end. Okay. So another thing that works to control actin, this is actually a toxic chemical. So phallotoxin or phalloidin is found in these death cap mushrooms. They're appropriately named because they're incredibly fatal because they are binding to actin and stabilizing it. So again, it's restricting this dynamic instability that allows the growth and depolymerization of these actin chains. Yeah, definitely don't eat any death cap mushrooms. This other molecule derived from mushrooms acts in a similar way. So I guess, again, to just highlight the importance of this dynamic instability in terms of the function of the cytoskeleton inside the cell. Yes. So it binds to it very tightly. So it's a multiple cyclic chain. It's not necessarily any electrostatic interactions. I'm not sure actually what the binding mechanism is there, but it binds very strongly to actin. And this is actually also really important for research. When we look at cells, we can stain them with this fluidin, and the fluidin molecules can come bound with a fluorescent molecule, and that fluorescent molecule allows us to visualize where the actin is because the fluidin is binding specifically to the actin. Okay, so two more proteins that are working in terms of the regulation of actin polymerization. These become especially important in cell motility. So again, at this leading edge, this is where the cell is growing or moving. So there's growth occurring in this direction because the cell is moving in that direction. And so ARP23 is one of these proteins, and the function of this is that it is creating branch points that allows for new nucleation sites. So you can see the green one, it's the green protein in this, and it's binding to an existing actin chain, and then a new actin chain can grow out from there. So this is just expanding the amount of actin polymerization that can occur at one point. And the WASP protein works with ARP2 as an activator. So once ARP23 and WASP are bound, then it enables the increase of nucleation sites. And this is just a electron microscope picture of what this looks like, a lot of interconnecting networks of these proteins, specifically at the edges of cells. All right, last 2 minutes, we're going to go through how Listeria works in leveraging these. So specifically, Listeria has a protein unit on one end, which the importance of it being on one end is key in terms of this propelling action. So it has this ACTA2 tail. What this ACTA2 tail has is it has this ARP2 activator, which allows, again, for this, the nucleation sites or the new polymerization of actin to occur. Oops. And this is occurring on the end of the Listeria bacterium. So it's bringing in all of these actin monomers and binding to this one end where ACTA2 is present of the bacteria, and it's enabling this force to propel it because of this polymerization that's occurring on the back end of the bacteria. And so you can see in this electron micrograph some of the actin polymers that are left behind as this Listeria bacteria moves through the cell. So a couple of quick treatments for listeriosis. Time is generally sufficient for healthy people. Even though these can move between cells, your immune system can still attack it, and we'll go through more details when we talk about the immune system. But for people with a severe infection, antibiotics can usually treat this, or general prevention is often advocated for. So avoiding some of these foods, like I mentioned, that are known to cause Listeria food poisoning. All right, and if you're interested in cytoskeleton or cell migration, a couple of interesting, great faculty at UVA that are doing this work. All right, any questions before we transition to the journal club? Cool, glad we made time. Hopefully you guys have enough time for the journal club. Again, I don't want this to be like a stressful activity. I think something to keep in mind as we're going through this is it's okay not to know. A lot of this information can be really overwhelming at first and can be probably way out of your comfort zone. And so part of this is just to get you comfortable with trying to learn how to address these things. How do you tackle this new information and draw something meaningful out of these research papers, especially as you work towards your project? So you might have experienced this already as you're going through the literature, looking for ideas for your disease application, that reading journal club papers can be really intimidating. So we just want this to be a useful exercise for you all. Again, a couple of guidelines, just as a reminder, it can be really helpful if you work individually and then bring your insights together, just maximizing the number of people in one team. Using the resources. So your teaching team is here to answer questions, but definitely try to seek out answers on your own first, because there are only limited number of us. And focus on the purpose of the experiment. So keeping in mind this hypothesis and the overarching goal of this paper, all of these figures should be supporting this hypothesis in some way. So in terms of trying to figure out what your figure means or what the purpose of it is, if you look back at these hypotheses, then they should be able to match to one of these directly. And as a reminder, this paper is about the extracellular matrix, is about fibronectin in particular, and this mechanoswitch that is known or thought to be known in fibronectin, where the different conformations of this protein can change the affinity for different types of integrins. And the argument in this paper is that some of those integrins might be playing a role in the development of fibrosis. So I included the link here if you want to look at the whole paper. Sometimes looking through the introduction might be helpful. And these two tools that they're utilizing throughout the paper will come up again and again. So if you have any questions about these, feel free to ask any of us. But you should all have access to the slides where all of the figures are put into the slides with the respective group numbers. So these are the groups with the 10-figure assignments. So it's going to take some moving around for this so that we can get into the respective groups. But let's go ahead and do like group 1, 2, and 3 on the side, and then 4, 5, 6, 7, 4, 5, 6, 7, and then 8, 9, 10. So hopefully you guys know where to go.